Oi, eu sou Marcos Yoshinaga, fundador da Penguins Lab, e hoje eu dei uma palestra sobre o SCIFLIX. Se você tiver curiosidade de saber o que é, fique atento aí no YouTube.
He he did his PhD in oceanography in 2007. Then he did a postdoc period for 8 years in Germany at the University of Bremen, where he specialized in the study of lipids from deep sea microorganisms. Then in 2016, he joined our group, where he helped us to establish the platform of lipidomic analysis. And then in 2023, he started he not started startups, strange in English. But so he is the CEO of Penguins Lab, a startup that is specialized in lipidomic analysis. And in 2024, he's got a position at Universidade Cruzeiro do Sul. So, and now he is doing very interesting work using isotopic labeled lipids to do tracing studies in cells and so on. So, thank you, Marcus, for accepting to talk to us.
Okay. It's a great pleasure to be back and talk about my work here. Before I start this talk, I need to do 2 disclaimers, and one is it has been a while since I gave my last talk in English, I think 5 years ago or something, so I might be mumbling a little bit. And the other one is that what I'm going to show you today is just an idea and not really like a concrete, group of results and papers and everything. So it's more like an idea than anything else. So don't be mad at me if you're expecting like very concrete results. The title of this talk is Stable Carbon Isotopic Flux Analysis by Lipidomics. And SCIFLIX addresses lipid metabolic activity. And thank you, Sayuri, for introducing me, so I'll just keep that part. And the content of this talk, I'm going to be presenting to you the ideation, where this idea came from, and then the rationale to use stable carbon isotopes for tracing lipid metabolism, and the multiple ways we can do this SCIFLIX. Also, the major challenge, I think one, or at least I think it is the major challenge for doing this, and then the resolution. And I'm going to show you 2 types of case studies. One, tracing lipid metabolism using labeled fatty acid, and the other one is with labeled glucose. And then finally, if I got time still, I don't know how long is this talk, but I'll show you some possible applications for SCIFLIX. So Sayuri explained really well my background. So I don't come from the field of biochemistry or metabolism. I did my first postdoc in Germany, studying the membrane lipids and stable isotopic tracing experiments in extremophilic microbes. And those varied a lot from Arctic to the deep ocean, but with temperatures of 2 degrees C to, 120. And then I got to know that, by the way, that photograph is inside a submersible called Alvin. And if you guys don't know Alvin, it was the submersible that discovered the Titanic. I think it was back in the 70s. And then by the end of my postdoc, I got to know this gentleman here. He passed 2 years ago, and he turned out to be my mentor in science. And he taught me a lot about lipids as unity for life, and then using my skills in mass spectrometry to look for lipids or lipidomics in health and diseases. And then I got back in Brazil, talked to Sayuri. I think I know Sayuri since 2013, if I'm not wrong. And then we discussed a little bit, and then she accepted me as a postdoc here. So I stayed here till 2023, I think. And then I made a lot of friends, but I think one of my best friends in science is Adriano, and he moved abroad in 2023. And then we were discussing about like what to do next and how could we come up with the idea of working together back again. And so this idea of SCIFLIX is mostly given to these interaction with Adriano. And then the rationale of using stable isotopic tracing is that by doing lipidomics, we know a lot about the concentration. So we know the abundance of the lipidome of a cell, which is more more or less like a picture. And then the stable isotope tracing, on the other hand, it gives us flux. And when you do like a flux analysis, then you know about the material flow from one end to the other. And I could explain that like as a photograph versus a movie, but I think Joshua, I stole this from Joshua Rabinovitz from Princeton, and he explains this flux analysis more or less like you're driving a car, and then the flux increases with car density until the traffic slows down by narrow road, and then you have finally very high car density, but almost no flux. So the status of a lipid in a cell equals the rate of synthesis, but also the rate of degradation. So it does not really tell us like the rate of synthesis or degradation at all. So using stable isotopes or stable isotopic tracing, it will be a way to probe this kind of stuff. And then the idea behind stable isotopes is that in nature, the heavy isotope, like a still stable isotope, carbon-13, it has an abundance of about 1.1%, and whereas the carbon-12 has an abundance of almost 99%. And so, given this distribution in nature, then and also given the one neutral difference between carbon-13 to carbon-12, which gives us about 1 Dalton difference, a substrate enriched in carbon-13 then becomes unfamiliar and natural, and then we can trace. And given this difference in mass, then mass spectrometry is the technique of choice, although you can use NMR to figure it out too. And so a lot of studies have been published on this matter, but everything that people look at is this mass shift from 1, 2, 3, and so on. And so you have natural abundance of like natural isotopic distribution, but then when you do a label experiment for, in this case, with uniformly labeled glucose, then you start seeing like the isotopal loads or masses higher than maybe 20 Daltons higher than the normal, the precursor molecule. And in terms of lipid metabolism, you can do that using a bunch of substrates. And I think the four most important for lipid metabolism is glucose, fatty acids, glutamine, and acetate. And as it shows here in this very simplified lipid synthesis scheme, but you can also use others like pyruvate and so on. But everything needs to be labeled with 13C. Not the whole molecule. You can, for instance, have like a very specific labeling of carbons in like a glucose molecule and I don't know, citrate molecule. But in our case, we use we prefer to use uniformly labeled carbon, or 13C. And a major challenge for this kind of study is like, take for example, glucose, labeled glucose. Although it's uniformly labeled, you have the cell has access not only to the labeled substrate, but also from the natural occurring glucose. And so when glucose enters the cell, then you might get like carbons in the glycerol 3-phosphate pathway to build like phospholipids and glycerol lipids. But it also might go through the whole thing here and then finally to citrate, which is exported from mitochondria and then becomes acetyl-CoA. Acetyl-CoA could, give the carbons needed for de novo lipogenesis. And in this case, in a pool of acetyl-CoA, you have both labeled paired carbon and also unlabeled or natural occurring pair of carbons. And then this stain produces a mix of fatty acid isotopologues, which can vary from 2 to 16 13C labeled carbons. So it's very complicated and it's not trivial to analyze. And so what we figure out in the literature is that the stable isotopic tracing with lipidomics is a very weird case scenario, because you have the ratio of review articles like pointing the potential of this technique to probe the metabolism of cells to the actual article showing data on this matter is very low. That means that you have a bunch of reviews and few articles showing real data. And then the other thing that by reviewing the literature we figured out is that most of the studies are using labeled fatty acids and not way more than labeled glucose, because it's straightforward to use lipidomics as a tool to analyze these things in a labeled fatty acid than it is in glucose. So how to solve this problem? We've been doing this effort since I started here at the biochemistry department in 2016, of trying to, at least for most abundant lipids, use manual inspection of MS-MS. And so by looking at MS-MS spectrum, we can identify molecules with a very a high degree of liability. For example, here in this chromatograph, you see 2 peaks with the same exact mass, and then 2 MS-MS spectra for each peak. So the peak on the right here, which is shown in a red area here, is this compound, and where we can see not only the molecular, the precursor ion, but also the fragments. And by doing like a match with the type and then the mass of the fragments, we can reconstruct the molecule and exactly point to the identity. And the same is true for this little shoulder here on the left. When you have a MS-MS spectra, then we are able to precisely identify the molecular species of lipids we're talking about. And so we've been doing this since 2016, and we learned a lot of stuff, but it's a tedious method, and takes time, and people get impatient about it, but it is very precise on the other hand. And so using this kind of approach, we did some studies with uniformly labeled linoleic acid. It has 18 carbons and 2 double bonds. And in this case, linoleic acid can be esterified like intact, like in this case, where you have here the mass 297 representing 18 daltons more than the normal linoleic acid in a compound called phosphatidylcholine. But also we can see the products of linoleic acid metabolism, like linoleic acid turning into arachidonic acid with 20 carbons and 4 double bonds. So 18 carbons from the labeled linoleic acid plus 2 carbons from unlabeled acetyl-CoA. And this is very trivial compared to this thing here. This thing here is when you label your substrate, or like you use 13C labeled glucose, uniformly labeled glucose. So the glucose can be anywhere, right? I mean, in this example here, I'm showing it. This is not the same compound as this. I couldn't find it, and I didn't have time to draw this thing, and can draw. So this thing here, this lipid here is a phosphatidyletanolamine with, well, here is 18-0, but 18-1 and arachidonic acid. And I can recognize here the fragments of 18-1 and arachidonic acid, but the change here in mass does not occur in the fatty acid chains, but occurs at the level of the glycerol moiety here. And how can I figure this out? Is just because normally phosphatidyletanolamine has 140 of mass, And then when you join the PE plus the glycerol, the mass becomes 196. And then in this case, using 13C labeled glucose, all of a sudden I see the mass 199, which gives us the exact mass of 3 carbons labeled with 13C. All right. And then this is another case studying a ceramide that has 3 hexacyl groups or 3 glucoses. And then normally the mass is 161 and 179 for each glucose. But then here I started to see the mass 167 and 185, which is a 6 Dalton shift from the original molecule. So then we can precisely say that this compound has a incorporated glucose, fully labeled glucose. And then the last example is that the label occurs not only in the glycerol moiety of phospholipids or glycerolipids, but also at the level of fatty acid chains. And then here is an example of an palmitate, which has 16 carbons being labeled with 6 of these 13C labeled carbons from glucose. The questions come at the end. How long do I still have?
20 minutes.
20 minutes. I'm not gonna be able
Or 15.
Okay, so here we go. I'm showing here a published study that was done in collaboration with Mikhail Shchepnov, Russian guy that is famous for this deep deuterated polyunsaturated fatty acid production. He just published a book I haven't read yet, but here's a little advertisement for his book. But what we did here in this study, we knew for a bunch of years that when you exchange 2 hydrogens here at the basolytic position of the carbon between the double bonds, then you get protection from oxidative stress, right? And then by doing this, it was shown, like since 2011, was shown in several different models that this D-PUFA really works like an antioxidant. But since 2011, these guys didn't know where this deuterated PUFA was going. And so the research question of this study here was real simple. We said, like, oh, let's give some deuterated linoleic acid to keratinocytes and see where it goes in the lipidome. And using deuterium, and actually a bit at the basolytic position, it's just 2 deuterium, so it's a 2 Dalton challenge. It can, we use mass spectrometry and we can recognize at a high resolution what is a D-linoleic acid from what is an oleic acid. Both have really the same, almost the same mass, and you can tear apart those things by doing mass spectrometry at high resolution. But one of the things that is amazing about these compounds is that the molecule that has the same mass, like the phosphatidylcholine 16081 in this case, and then the phosphatidylcholine 160D linoleic acid, they have they come in a chromatogram at different retention times. So it's very easy not only to follow this trend here of differences in mass, but also in differences in retention time, which facilitates a lot the quantification of these compounds. And so in this study, we identified and quantified 309 total lipid species. 206 of those were composed of natural isotopes, and then half of that, 103 molecules contain at least 1 chain of D-linoleic acid. And then by doing lipidomics, we can actually obtain like a quantitative data, and then we figured out that the incorporation of D-linoleic acid to the total lipidome is about 20%, and we can, do this kind of plot where you find that most of the D-linoleic acid was going to the structural lipids like PC, which are major lipids, and PE, and a bunch of that, almost a fourth, was going to the triacylglycerides or TGs. But then we can also look into this data qualitatively, and we figured out that 78 to 68% of the storage lipids in the form of TG and ADG, which is like a storage form for ether lipids. A bunch of those were labeled with or contain D-linoleic acid. But then after that, but surprisingly enough, right, because cardiolipin and phosphatidylglycerol, they are not major lipids at all, and they do not contain a bunch of these D2-linoleic acid. They had like a very significant part of their molecules of these two subclasses enriched with D2LA linoleic acid. So we figure out that storage and mitochondrial lipids were the most important sites for esterification of this kind of fatty acid. And so the research question was answered, and the major findings was that the D-PUFA, they go in keratinocyte at least to triglycerides and storage molecules, storage lipid molecules, and then a bunch of those go to the mitochondria. And there's a bunch of hypotheses as to whether these compounds act as an antioxidant in mitochondria. So we just went one step ahead to prove it, that these things, they go inside the cell and maybe they go significantly into mitochondrial lipids, phosphatidylglycerol and cardiolipin. Okay, this other study with fatty acid was performed by Adriano and Thiago. Thiago is here, Adriano is in Germany. And these guys were after, well, in the umbrella of Professor Sayudi's lab. And these guys were after the inhibitors that may change the way ferroptosis acts in the cell. Ferroptosis is a kind of regulated cell death caused by accumulation of ion or defects in antioxidant machinery and enzymes like the glutathione peroxidase 4, which is responsible to detoxify some lipid peroxides. But anyway, I'm not going to talk about too much, but the results that they found out using these inhibitors is that, well, it's very well documented in the literature also, but the fatty acid saturase S2, it really played a role by not letting the cells die like the control with the MSO and the other inhibitors. I'm not going to talk about them, but they didn't even scratch the surface in terms of effect. And so we wanted to study what happened when you put this fatty acid saturating inhibitor in the cell. And this is what we figure out. How's my this is what we figure out. I construct this plot here with the circles here represent real abundance, so they are comparable among each other. And then within the membrane lipids, we kind of construct like a pie chart with the contribution of each subclass of phospholipid. So in order to understand this graph here, you need to pay attention to this. Then the storage lipids are basically triglycerides and cholesterol esters. They are in a gray scale here, gray black scale, whereas the membrane lipids are in the color scale. And then you have in the center the labeled linoleic acid, and then a linoleic acid can be the saturated. You can add double bonds to it, and you can also elongate it, like adding carbons. Whereas here to the left is just the action of the elongases, putting some more carbons into the linoleic acid 2 by 2. And this is what happened. In the normal situation, the 13C labeled linoleic acid, a bunch of that pool that you put in the cell goes into phospholipids. And in this case, the phospholipids already desaturated by this, also by the fatty acid saturase number 2, but also fatty acid saturase number 1. But anyway, the pool of PUFAs here, or polyunsaturated fatty acids, are huge in phospholipids and also as a cholesterol ester. Then when we use like a glutathione peroxidase inhibitors, like the RSL3, then the picture changes a lot in that you have way less polyunsaturated fatty acids into the membrane lipid pool. I used to have a bunch of C here, the cholesterol ester, but then you see this shift accumulating ATP or linoleic acid as it is in the membrane lipids, and mostly in PC. And then here you see the shift that PE was, it almost disappeared from the pool of phospholipids. And then when you use the fatty acid saturates to inhibitor, you kind of get the same thing as you inducing fructosis, but the pool of PUFAs into phospholipids get really low because you're inhibiting this pathway here, and everything gets accumulated here as it is, right? The 13C linoleic acid in membrane lipids and very few storage lipids. But then when you use these two combined, then you have a picture that is more similar to this situation here. And this thing here prevents the cell to die. And the difference between this pattern here to this or this or this is that we lose a lot of phosphatidylethanolamine, PE, but also some important lipids like cardiolipin. And so tracing with 13C linoleic acid and using fatty acid saturating inhibitors, it shows more directly how the dynamics of polyunsaturated fatty acid works in ferroptosis. The evidence or the putative evidence that people believe is that also phosphatidylethanolamine, but the species linked to arachidonic acid or adrenic acid get peroxidized, and this causes the cell death by ferroptosis. And then we put a little spin on it, also evidencing the role of phosphatidylethanolamine, but now putting cardiolipin as also a probable target of ferroptosis. And I forgot to mention, but this work here was performed in glioma cells. Do I have more time? 10 minutes.
10 minutes.
Wow. Okay. So this case study was performed with uniformly labeled glucose. And then we used a cancer cell called the renal cancer, clear cell cancer cell, something like that, number 4. And what is interesting about this cell is that it has no VHL, which stands for von Hippel-Lino something. And this gene is responsible for the HIF or HIF activation. And that means that these guys here in a natural constitution with deleted VHL, they have like the sensor for hypoxia activated. So the cells feel hypoxia the whole time, and so the cell becomes glycolytic. And EV stands for empty vector, and VHL is where they put the VHL back into this cancer cell line. And then when you put the VHL back in the cell line, then the cell starts to sense oxygen, and that's fine, and then it becomes oxidative. It's not a model of cancer versus no cancer cells, but it is a model, a very good model of glycolytic versus oxidative kind of metabolism in a same cell. They performed some metabolism measurements here with seahorse, and to prove that the VHL was more oxidative, also performed some target metabolomics, and then they figured out that in the glycolytic cells, there were accumulation of the big products and also the substrate, glucose, pyruvate, and lactate, whereas in VHL, you have accumulation of the intermediate. And so we treated the cell with uniformly labeled glucose for 48 hours, and then performed lipidomic analysis. And the thing about this project, which is very special to me, is that we didn't even change the routine of analysis that we do normally in lipidomics. So we did some manual identification, produced a list of ions with very defined head group coupled to the fatty acid chain, and then we send it to a software called Maven, and then the Maven got this list, produced a bunch of isotopologues from this list, and then we used the isocorrector, and is an ARC script, just to trim out the natural abundant isotopologues. And so it was kind of simple in this sense, but yeah, it's kind of complicated, but it's simple. And the rationale here was that by using uniformly labeled 13C glucose, we would be able to see the labeled carbons coming from the glycerol 3-phosphate, right, with 3 carbons, so odd kind of number of labeled carbons, and also the acetyl-CoA here with 2 carbons, 2 labeled carbons, meaning that everything that comes from acetyl-CoA will be like even in terms of real number of carbons. And then we track this distribution in the lipidome. And we initially identified the top 150 most abundant lipids, which turn out to be like, at the end, 4,000 isotopologues, different isotopologues. So it introduced a very challenging quantification procedure, which we've done mostly manually. And so it's a lot of work. And then with that, quantifying 4,000 lipid isotopologues, and we come up with this distribution here. The circles again are in the same unity or of abundance. So there's a little bit more lipids in the VHL, but what is astonishing about this data is that the percentage of labeled carbons in these lipids is higher in the VHL or the oxidative cells than in glycolytic. Just that for the glycerophospholipids and also for the sphingolipids. And then we did some subclass analysis here with a bunch of subclasses, and we shown that most of the times, most of the cases, the glycolytic or the oxidative VHL has way more label than, or significantly more label than the glycolytic EV. But in some instances, like here, PPC and PPE, which are both plasmalogens as kind of ether lipids, then the glycolytic was incorporated more label than the oxidative. Okay. And then if we were to put every single lipid precursor and the distribution of isotopologues in the graph, it would look like this, right? Where you can see here, here is the fraction abundance of the precursor, n plus 0, and then you start 1, 2, 3, and then goes to 30 to 40 isotopologues. And from this graph, it was already very visually or clear that the oxidative PHL cells, they produce longer or heavier isotopologues, like the n plus 13, 10, blah, blah, blah. Whereas the glycolytic, they were more focused in the shorter n plus 1, 2, 3, 4, 5 at most. But it was like that. You need to believe that it was like that for almost all lipids, including sphingolipids and some other prenol lipids like the 10. But the challenge here is like you would have to plot one of these for each one of the 150, and then you end up with the 150 plots, and it's not feasible. So how to display the data? And then we come up with this graph, which is very hard to interpret, but I'll try to explain to you. These are distribution of phosphatidylcholines. Those are the species I'm talking about. And these instances here, this graph shows like a fold change in the comparison EV versus VHL. So these lipids here are, or these isotopologues of these lipids are more abundant than the VHL in EV than the VHL. And here is the opposite, the ones that are more abundant in VHL than the EV. And what is clear is that the lower mass isotopologues like until n plus 5 are sometimes higher in concentration than in the glycolytic than the oxidative, whereas the higher masses isotopologues are more concentrated in the oxidative versus the glycolytic. So that was one thing that we had to do to interpret the data. And then what I wanted to show with this plot was the isotopologues. I divided them by even and odd. Odd meaning that most likely the label was in the glycerol moiety rather than in the fatty acid because of the glycerol 3-phosphate. And then we show that independent whether it's even or odd, the oxidative cell always had the most abundant concentration, whereas it only occurs in the plasmalogens that, and in the odd isotopes that the glycolytic one. So again, evidencing the important role of de novo synthesis of plasmalogens for the glycolytic cell. And then here I said, like, okay, then let's limit to the lower masses isotopes, like M+3 and M+5. And then we saw, like, okay, now the glycolytic really changed the game, right? Everything is more concentrated in the glycolytic, except for PE. And then we said, like, okay, this lower masses only represent the glycerol, maybe try to make like a proportion of odd isotopes to the total labeled molecules. And then here we go. We figured out that a bunch of cardiolipids were more enriched in terms of concentration in the oxidative versus the glycolytic. Okay, so here is what we think of a hypothetical model where you have in the glycolytic cell a bunch of carbons from glucose going in this direction, or glycerol 3-phosphate, and also this other molecule that is used for the synthesis of ether lipids, and then that's why ether lipids are more enriched than in the glycolytic cell. And that was sort of more or less expected, right? That the glucose will be used here because of the majority of the glucose will go out of the cell as lactate in the glycolytic. But then in the VHL, which is oxidative, then glucose goes all the way to pyruvate in the mitochondria, then citrate, acetyl-CoA, that goes into lipogenesis, de novo lipogenesis, and then fatty acids get a lot more labeled just because the glucose goes all the way here. And some of the surprises was this higher concentrations of labeled ether lipids, plasmalogens in this case, and this may point to the role, an important role of plasmalogens in cancer cells. And this higher concentration of typical mitochondrial lipids or the oxidative VHL was kind of very surprising because Q10, PE, and cardiolipin are often not the most abundant lipids in the cell lipidome. But you see, like, even though they are not the most abundant, the flux of carbons from glucose to these lipids is very important for the oxidative cell. And I'll go through real quick. This work was done in collaboration with Cynthia Portugal and Hiro Goto. And what we investigated was a model of parasite and macrophages and in Leishman and Infantum kind of infection. And then the results were mostly related, like the major fold changes here. And when you compare infected macrophages to the control were related to the lipids from Leishman and Infantum. And you gotta believe me because this graph is just a bunch of lipid classes here. But other than that, we found some lipids that we could not really tell like who was producing this kind of thing. It could come from the parasite, but also from the macrophage. And well, by using this combination of results, we figured out that the LDL was actually playing a role. Uptake by macrophage, so the LDL is uptake by macrophage, and then becomes endocyte, becomes an endosome, and then instead of fusing with the macrophage lysosome, it was fusing with the parasite vacuole. And then by fusing here, then in this acid milieu here that is found in this parasite vacuoles, then the LDL will be degraded into free fatty acid, free cholesterol, then will serve as the substrate for the lipidome of the leishmania. And one thing that I forgot to mention here is that a bunch of these cholesterol ester, free fatty acid, and triglycerides that were enriched in the infected macrophage were linked actually to polyunsaturated fatty acid and what we call with 2 and 3 double bonds, and then also 2 highly unsaturated fatty acids with 4 or more double bonds. And so by reading the literature, we saw that leishmania has a very specific desaturase that can transform, for instance, oleic acid, which is an 18-1 in 18-2, and this we mammals cannot do. And then you can go further and transform this 18-2, which is a linoleic acid, into 18-3 omega-3. So you can do a bunch of stuff with the leishmania desaturase machinery. And so we haven't proved nothing yet, but it's a very descriptive kind of study. But in order to prove this kind of stuff, this kind of transport, and also this kind of lipid modification by the desaturase, we could definitely use a 13C labeled oleic acid and put it into an LDL-like particle, and then let the system run until we get these labeled PUFAs and eventually highly unsaturated fatty acids. Okay, the last one is a project that we have submitted as a scientific initiation kind of fellowship. And this again comes from data from Sayuri's lab, but who performed these experiments was Thiago. And here what he did was to use the C6 glioma model and then use RSL3 as a ferroptosis inducers, and then different concentration of 2 fatty acids. One is arachidonic acid, which has 4 double bonds, and then oleic acid, which has just 1. And then the fact that really matters is that when you put oleic acid, independent of the concentration, you sort of protect the cells against ferroptosis. And when you do that with arachidonic acid, you enhance you get more cell death than in control. And so the arachidonic acid is like a ferroptosis inducer by itself, because it gets esterified into phospholipids. But then when you use a mixture of this oleic acid with arachidonic acid, and then you kind of protect the cells against this death caused by enhanced concentration of arachidonic acid. And so there's a lot of information in the literature about the role of oleic acid as an antioxidant in different sort of models and conditions, also in ferroptosis. And the main explanation is that when you put oleic acid into cell culture, the polyunsaturated fatty acids, they do not get esterified into the phospholipids, rather they go into the storage lipids. But these are all like very elusive kind of hypothetical explanation. And so by using maybe a 13C oleic acid, we could then figure out where actually the PUFA go, or whether the oleic acid is influencing the esterification of PUFAs and everything. So here, I'm coming to an end here. SCIFLIX has a lot of limitations. One of the most incredible limitations is the amazing price of uniformly labeled glucose. It's very expensive for basic research, but it's basic research, right? So studies that use in vivo models are really limited by the cost of the whole experiment per se. And then the other thing that's why I said it was an idea, is that I don't have like a multiple sampling points that you could use a time course to calculate flux analysis, to do a flux analysis. And so this is something that we could improve, and we will improve in the future. And I think also one limitation is that we study a very specific model of renal cancer, and then all we want to do now is to extend that for multiple models, because we need to test the viability of this method to in other settings. Anyway, the take-home message is that flux analysis may significantly impact the field of metabolism, and any future work requires precision in the identification of molecules in order to get these isotopic loads really well quantified. Here, I want to acknowledge the funding agencies. A lot of this work wouldn't be possible without the Professor Sayuri and her lab. Dr. Adriano is my partner in science still, and Professor Almushutz they are all from the KFZ, the Center for Cancer Research in Heidelberg in Germany. And Professor Only was essential because when we were talking about this glucose experiment, she was like, oh, so yeah, I got your point, but what's the question? What is the big question? And so I had to explain to her that we wanted to do like these experiments as a technological development kind of stuff, and not really like apply to something or with a big question. I have a bunch of collaborators that I should mention. I might have forgot some. You can find me at the Penguins Lab, both in the website and Instagram or the other platforms. We recently launched the Liponautas and Lipidicamente, which is a branch of the Penguins Lab that will be talking about more about science dissemination, because I'm very research focused, and then these ones were created and now financed by capacity to be disseminating ideas and research on lipids. Okay, I thank you all for the attention.
Thank you, Marcus. Let's open for questions. I'm not sure. Any questions from the online people?
I don't think so, Sayuri.
No, too difficult, perhaps. Any questions here? Alicia?
So this is almost philosophical. How do you distinguish the chicken from the egg? For example, in your cancer cells, in which you have HIF activated or not, and then you see that when you have more oxidative phosphorylation, you have more cardiolipin, and you also have more production of fatty acids from the glucose going to citrate. Is this because there's more mitochondrial ability to generate citrate, or is it the opposite? Is that you have these lipids because of the HIF factor, et cetera? How do you distinguish what comes first?
I have no idea how to really distinguish the chicken and the egg, but what I can tell is that even though we cannot really distinguish which generates what, I think that this work really shows and illustrates how is the dynamic of mitochondrial lipid production when you are oxidated. And this is something that in the lipidomic data, without tracing, impossible to see sometimes. You see very discrete fluctuations in, for example, cardiolipin levels or PE or Q10, but never had this look into the cell saying like, okay, then they use mitochondria and might have to, rebuild the whole time those lipids because they got in, oxidized maybe.
Have you ever thought of looking at the proportions between these lipids and mitochondrial protein markers and their turnover? Because that could give you an idea.
That's true. We only focus in lipids by now, but yeah, that is a great idea. We should definitely try to do that.
I think it was quite difficult, perhaps, because too much of mass spectrometry data. But one thing I thought was very interesting is the fact that in the glycolytic cell, when you give the labeled glucose, you could see an increase in plasmalogen species. This increase was due to the labeling because you showed two parts. One going through glycerol, 3-carbon enrichment, and the other going through glucose being converted to acetyl-CoA, 2-carbon, and then you will label the fatty acid. I could not follow well if you could see the plasmalogen increase through glycerol or through acetyl-CoA. Where was the labeling when you showed the plasmalogen increase?
So there were two pieces of data that I showed, but it's very complex. I admit, very complex. But anyway, what I showed was like in odd isotopologues, like in this case here, not the even, but the odd isotopologues, there was an enrichment in plasmalogy.
When you say odd,
Odd
it's
is-
because it's glycerol label.
Exactly.
The glycerol is,
It's the,
so you were seeing, okay.
but not in the even ones, not for all species in the even. And so I think a pathway for plasmalogen synthesis is really really through the glycerol 3-phosphate, one from glycerol 3-phosphate to DHAP, and then to peroxisomes to deal plasmalogen.
Yeah.
Like I pointed to.
Okay. Because usually think in the phagoptosis context, the glycolytic ones are more resistant to that, yeah? I think the literature says that, if I'm not wrong, I don't know, No, I don't know if Adriano showed it. Anyway, because this could be one of the explanations. If you, in the glycolytic one has more ether phospholipid, which is antioxidant, usually can work as an antioxidant for cancer cells. So
The more they have, the less susceptible to.
yeah, that was something I was thinking, looking to this data. The glycolytic cancer cells use this glucose to synthesize more plasmalogen. But yeah, that's one of. Yeah. So I think it's already time. Thank you very much, Marcus. I think this shows that this is the beginning. Just to be keep it clear here. This is the beginning of using this approach, the isotopic labeling, to study lipid metabolic flux. So I think Marcus did showing a picture of the beginning of this year. So thank you, Marcus, for your excellent help.